Welcome to Lecture 3 of our Hereditary Genetics, Unit 7. Okay, so let's talk about some basics of heredity genetics. So, for diploid organisms that reproduce sexually, an organism is going to have two alleles for most traits. It's going to be one from each parent. So when that organism reproduces, it's only going to pass on one of those alleles for the traits. Because again, one comes from mom, one comes from dad. So the different variances for a gene are called alleles. So basically what that means is that an allele is a different variance for the exact same gene. So for instance, in pea flower color, purple is going to be dominant to white. So since purple is dominant, that means that white is recessive. So here you can see that we have the chromosomes. Here are the allele for the color of the flower. And you can see that we've got purple and we've got white. So when we look at that, let's say that this is from dad and this is from mom. Since the P is dominant, it would be a capital P. And when we're deciding to label colors or label traits, when we're looking at those, we always go off of the dominant trait. So in this case, it would be P. So for white, since it's recessive, it would be a lowercase p. And it's always a good idea on the lowercase, especially when it's difficult to tell if it's capital or not, to put that line over it, because that tells you, make sure that you understand that it is lowercase. So in this instance, we would have a genotype of capital P, lower p. And we would have a phenotype of purple. Because again, the purple is dominant to the white. So Gregor Mendel is the guy that came up with all of the ideas that we just talked about. He's also called the father of genetics. And what he did is that he basically tried to figure out how things were passed on, how traits were passed on. So he took pea plants and with different phenotypes. He recorded those phenotypes in the offspring and determined the following ideas that are termed the idea of Mendelian genetics. So what he said and what Mendelian genetics is that each person has two factors for every trait. The two factors will segregate, and only one of those factors is passed on to the offspring. This is referred to as the law of segregation, which is also listed right there. So the passing on of a factor happens independently of everything else, all other factors. And this is called the law of independent assortment. Each offspring will have one factor from each parent. One factor is going to be dominant over the other. And this is referred to as the law of dominance. So it's important to note that Mendel only worked with traits that show complete dominance. So he didn't know about any other types of inheritance like codominance, sex linkage, any of those things. So those traits that we're going to talk about, they do not follow Mendel's ideas. It's also important that you need to make sure that you know the ideas of Mendelian genetics, and that you know the 3 laws of inheritance. So let's talk about the crosses that follow Mendel's ideas. So a monohybrid cross shows complete dominance. So these are traits that have complete dominance, and they basically, what that means is that they have an allele that can mask or cover up the other allele. So the allele that is expressed is going to be called the dominant trait, and the trait that is masked is going to be called the recessive trait. So again, we're going to use the first letter of the dominant trait capitalized to represent the dominant trait. So for instance, if purple is dominant to white, then the capital P would be for purple, and the lowercase p would be for white. So here, let's take a look at these different kinds. So we've got here 2 capital P's, which means we have 2 dominant traits. So this is said to be homozygous, meaning same. So it's going to be homozygous dominant, or purebred for purple. So the phenotype we're going to end up with would be purple. Here we've got a capital P and a lowercase p. So this is going to be referred to as heterozygous, meaning different. It can also be referred to as a hybrid. And our phenotype here, since we have a capital P over a lowercase p, will once again be purple. Here we have 2 lowercase p's. This is referred to as homozygous, again meaning same, recessive. It can also be referred to as homozygous white, but I will tell you it's usually not used that way. Phenotype, we are going to end up with a white flower. So one of the tools that we use with these Mendel genetics and the crosses are called Punnett squares, which you're probably familiar with, hopefully. But Punnett squares, they're going to indicate the alleles each parent can contribute to the offspring and what those combination of alleles would be. So we're going to look at a monohybrid cross, and this shows the offspring phenotypes that can occur for one trait. So remember that for a trait that shows complete dominance, tall is going to be dominant to short in these instances. So this says cross a homozygous tall plant. So since it's telling us a homozygous tall plant with a purebred short plant, so the first thing that we need to do is assign our letters. So we know that tall, capital T, is dominant to short, lowercase t. So a homozygous tall plant, homozygous meaning same, so that would be T, capital T, to a purebred short, so that would be lowercase t. Could also be referred to as a homozygous recessive. So that's our first cross, and what we're going to do is we're going to put one trait here, one of the genotypes here and one genotype here, and then we cross those. Here, cross 2 plants, both heterozygous for height. So what that means, heterozygous meaning different. So we've got 2 with a capital T and a lowercase t. Okay, and we're going to cross those, T and T, T lowercase t, and here we end up with 2 lowercase t's. Cross a hybrid plant with a short plant. So hybrid is another word for heterozygous. So we've got capital T, lowercase t, with a short, which we know is homozygous recessive. So those crosses, we are going to end up with these genotypes. So now that you've got the information for the crosses, they are going to ask you for different frequencies and ratios. So for instance here, a genotypic frequency. We have got 4 big T, little t. So our frequency would be 4 big T, little t. Our ratio would be 4 big T, little t. Phenotypic frequency, we would put 4 out of 4 will be tall. And then our phenotypic ratio, again, 4 tall. Okay, so now let's look at the next one, which has a little bit very different variations. So the genotypic frequency, what they want from you is they want to know that I have 1 out of 4 that is a capital T, capital T. And I have got 2 out of 4 that is a big T, little t. And then I've got 1 out of 4 that is little t, little t. So the ratio, you're going to do, it's the same thing, but you're going to do 1 capital T to 2 big T, little t, to 1 big T, little t, little t. So I want you to make sure and note these are important right here when you're doing ratios. Those are very important. So our phenotypic frequency, remember phenotype is the physical appearance. We're going to have 3 out of 4 that are tall and 1 out of 4 that is short. And our ratio would be 3 tall to 1 short. I want you to pause a minute and see if you can come up with the genotypic and phenotypic frequencies and ratios on this last one here. Okay, so what you should have come up with is 2 big T, little t, and 2 little t, little t. Or you could do 2 out of 4 on those. On our ratio, should be 2 big T, little t to 2 little t, little t. Our phenotypic frequency, we're going to have 2 out of 4 that are tall and 2 out of 4 that are short. Our ratio, we have 2 tall to 2 short. So that is the basic way to do Punnett squares. Something else that is important, especially for the AP exam, is that you should practice enough of these Punnett squares until you know the expected ratios and frequencies. So what I mean is, for instance, if your first generation, your P generation, is a cross between a big T, little t, and 2 big T's, then you should be able to figure out that you've got a 1 to 0 ratio. Okay? They're all going to be dominant. if you're looking at your P1 generation as a big T, little t, crossed with a big T, little t, then you are looking at a 3-to-1 ratio. Okay, you can see there's about 3 times as many. Because remember, that last box, that last square will always be recessive. And then finally, if you're looking at a big T, little t cross with a little t, you are looking at a 1-to-1 ratio. They're going to be about the same. So let's take that information and look here at an example. When a tall and short plant were crossed, approximately 151 of the offspring were tall and 142 were short. 2 of the tall F1 offspring were crossed, and it resulted in an F2 offspring. 156 were tall, while 53 were short. Determine the genotype of the tall plant in the P1 generation. So if we look at our generations, the first cross was pretty much a 1-to-1, but then we ended up with a 3-to-1 ratio, 156 to 53. So that tells us, based on what we just discussed, that the first cross, we're looking at a heterozygous and a homozygous recessive cross. So, test crosses, these are going to be done to determine the genotype of an unknown. So, the test cross in which the unknown is crossed always going to be crossed with a little t, or a homozygous recessive, okay, individual. You're going to cross the unknown with the known homozygous recessive individual and then analyze that offspring to determine the genotype of the unknown. So that tells us that the unknown is either going to be a big T or a big T, little t. So for example, if a pea plant is tall, our possible genotypes are going to be big T or big T, little t. So scientists are going to choose a plant that is little t, or homozygous recessive to help determine that unknown. If all offspring end up tall, then we know that the unknown likely is going to be a big T, homozygous dominant. If we end up with half tall, half short, then we know that the unknown is going to be a hybrid or a heterozygous. So let's look at another problem. Let's do a test cross. The tall offspring of the F2 generation could either be big T, or little t, big T. Discuss a way we could determine the genotype of a tall plant without sequencing the plant's DNA. So what we are going to do is we are going to do a test cross. We're going to take an unknown times little t, and then through that determine if unknown is homozygous tall or heterozygous tall by looking at results of cross. Okay, so in this case, they're not asking you to cross anything. They just want you to discuss it. So let's talk about some exceptions to the Mendelian genetics. So these type of inheritances do not follow the rules. Do not. So they include traits that show incomplete dominance, codominance, there could be multiple alleles, there could be sex linkage, linked genes, lethal traits, traits that are polygenic, and traits that are non-nuclear, which includes traits due to mitochondrial and chloroplast DNA. So, and the chloroplast and the mitochondria, because they're randomly assorted to the gametes and the daughter cells, so the traits determined by them don't follow the rules. So if a short answer tells you that you need to discuss a trait that does not follow the Mendelian genetic rules, pick one from the above and the traits that show the epistasis and the pleiotrophy. If it says anything about that, that's not your answer, okay? Because those are beyond the scope of AP. So we're not going to worry about those. These are the ones that we're going to worry about. But if you see those words and we didn't learn about them, then don't worry about them. It's not your answer, and just go from there. So let's talk about incomplete dominance. Basically, this is an inheritance pattern where the heterozygous genotype is going to produce a middle or intermediate or even blended phenotype. So we're going to have to use a different way to symbolize the alleles for that incomplete dominance. So we're going to use a capital and a prime. So an example would be like the snapdragon flower color. We've got red, we've got pink, and we've got white. So red, we're going to do R prime, R prime. No, I'm sorry, that's not correct. Red, we are going to do R, R. Pink, we are going to do R prime, R prime. Okay? So let's look at this. And let me go back up here and correct this. My apologies. The pink would be R prime, R, R prime. The white is R prime, R prime. I apologize for the typos. Okay, another example is hypercholesteremia. We are going to use the H, capital H, for low cholesterol, and we are going to use the H prime for high cholesterol. So here we've got homozygous for low cholesterol, we've got big H, that's low cholesterol. Heterozygous for hypercholesterolemia, we've got HH prime. This is moderate. And then we have H prime, which is high. So these traits are going to be exceptions to the Mendelian genetics because they do not follow the law of dominance. So let's look at some examples here. If we're going to cross a red flowering plant with a white plant, we're going to cross two capital R's with an R prime, R prime. If we went across two pink, then that means that is an intermediate. So we are going to cross an R with an R prime. And again, an R with an R prime. If we're going to cross a white flower with a white flower, then we would be crossing an R prime with an R prime. And then this is what we end up with. So if we look at our phenotypic frequencies and ratios, we will see that this first, crossing a red with a white, we're going to end up with all pink. That's going to be our intermediates, codominance. So our ratio would be for pink. If we look at our cross between two of the pink plants, Our phenotypic frequency would be 1 red, 2 pink, 1 white. So our ratio would be 1 red, 2 pink, 1 white. And here when we cross a pink with a white, which I just noticed my mistake, I apologize. Up here we're crossing a pink with a white, so I've got the genotypes wrong there. My apologies. So if we cross that pink with the white, then our phenotypic frequency, we will get 2 pink, 2 white. So 2 pink, 2 white. So let's look at problem 3. Identify the genotypes of the P1 generation for each of the following F1 generations. So this again goes back to our ratios. Here we've got 237 pink and 245 white. That is basically a 1-to-1 ratio. So we should be able to figure out that with that 1-to-1 ratio, we are crossing pink with white. So we are crossing that R prime with R prime, R prime. Here we have 488 red, so again, that's our 1. So this one, we're going to cross red. So we're crossing RR with RR. And then here we have 222 pink, 105 white, and 112 red. This is actually a 2-to-1-to-1 ratio. So with the codominants, this would be a pink RR prime crossed with an R prime. And feel free to go and do these crosses. Feel free to do the crosses so that you can see exactly how the ratios work, because it's important that you understand those ratios. Next we're going to talk about codominance some more. So this is the type of inheritance where we have got both alleles being expressed or seen fully in the heterozygote. So we're going to use 2 different capital letters for the 2 alleles. So for an example, would be a blood group. So a red blood cell with an M molecule on the surface, and then that would be MM. MN would be a red blood cell with both the M and the N molecule, and the NN, which would be the red blood cell with an N molecule. Another example would be a Rowan coat color. So you've got some with red hairs, you've got some with red and white, you've got some with white hairs. Another example is sickle cell anemia. So for instance, hemoglobin A leads to normal hemoglobins, hemoglobin S leads to abnormal. So let's look at that. Here we've got the hemoglobin A, so it's homozygous, and this contains normal hemoglobin. It's never going to be sickle cell. Here we've got hemoglobin A and hemoglobin S, so it's heterozygous, and the red blood cells are going to contain both the normal and the sickle cell hemoglobin. And in this case, it will sickle in low oxygen. Then we have hemoglobin S and hemoglobin S, so it's homozygous for that sickle cell. And the red blood cells contain only the abnormal, and these are all sickled, and usually it's fatal. So these traits are exceptions to Mendelian genetics because they do not follow the law of dominance. Oh, sorry. Okay, so let's talk about multiple alleles. This is when a trait is going to show, it's going to show multiple alleles when more than 2 alleles exist for the trait. So for example, a blood type. There's going to be 3 alleles for ABO blood type. So alleles might be completely dominant, incomplete dominant, or codominant to one another. So for instance, in the case of blood, A and B alleles are going to be dominant to O. Also, it's important to note that A and B alleles are going to show codominance. So if we look at the genotypes of these, this here, this is going to be A blood. This, it doesn't have a second marker, so it would also be A blood because it's dominant. Here with the A and the B, we're going to end up with AB blood. Here with 2 B's, we're going to end up with B blood. Here, because it's dominant, we're going to end up with B blood. And then if we have 2 of the recessive, that is our O blood. So these traits are exceptions because there's more than 2 alleles for a trait, and the A and B alleles do not follow the law of dominance. Now we're going to talk about polygenic inheritance. So this is when a trait is going to be controlled by more than one gene. So for example, we've got human skin color, eye color. If that trait is going to show polygenic inheritance, there's a continuous variation and there will be multiple phenotypes that result. So this example is wheat seed color. So there's 3 genes that control the wheat seed color, and each dominant allele has a quantitative effect. So in this case, just this graph, you can see that this is the light and this is the dark end as far as color goes. Then we have sex-linked inheritance. And this is the inheritance that relates to the genes on the X and the Y. So as you should know, females are XX and males are going to be XY. So these chromosomes are not homologous. They don't have the same genes. So for instance, the X genes, they have, they can code for muscular dystrophy, red-green color blindness, hemophilia. The Y genes carries very few alleles. It carries the spermatogenesis gene, the sex determining factor, ear hair distribution, as interesting as that is. But these traits are also exceptions to Mendelian genetics because males do not have 2 alleles for each trait. And the genes on the X chromosomes are called X-linked. And the genes on the Y are called Y-linked. So alleles that are written, for instance, XB, if the gene is located on the X, and it would be written YR, whatever this would be if the gene is located on the Y. So let's look at an example like color blindness. So the gene is on the X chromosome. Seeing color is dominant to color blindness. So if we have a heterozygous female with normal eyesight, she would be XB, XB, because she's heterozygous. If we have a homozygous female with normal eyesight, then we are looking at X, capital B, X, capital B. If we have a homozygous colorblind female, then we are looking at X lowercase b, X lowercase b. If we have a male with normal sight, we are looking at X capital B and Y. Colorblind, X lowercase b and Y. It is important that you know that conditions due to X-linked genes are going to affect males more than females. And if you think about it, really it makes sense because a female is XX, a male is XY. So the male only has one X chromosome. So basically, he's automatically going to be affected if he gets one recessive allele. There's no second X to hide or mask that recessive trait. So females will likely have a dominant allele that can mask the recessive allele. So for example, in fruit flies, the eye color is X-linked. So red eyes, X plus, are the wild type and are dominant to white eyes. So when you see, for instance here, wild type, you may make a note of that. That is usually the most common. And then when you see this recessive, that is usually mutation, which is usually X negative. So let's write those down here. So we're going to cross a heterozygous female with a wild type male. So we have heterozygous, so X plus, and we have an X minus with male, wild type, so that's X plus Y. And we're going to cross that. So here we're going to end up with X plus, X plus, X plus, X negative. Here we're going to have an X plus and a Y. And here we're going to have an X negative and a Y. So phenotypically, we're looking at two females that are the wild type, one male that is a wild type, and then one male that is white. Here we're going to cross a purebred wild type female with a mutant male. So we're going to end up with X plus crossed with a mutant. Remember I said mutant is the recessive, the white, so that would be an X negative Y. So if we cross those, we're going to end up with an X plus, X negative, X plus, X negative, X plus, and a Y, and X plus, and a Y. So we're going to end up with two wild females and two wild males. You must be able to analyze the results and determine the genotypes of parents used in crosses and the type of inheritance associated with that gene. In order to do this, there are two main decisions to make. And this is very similar to pedigrees. Hopefully you're familiar with pedigrees. If not, we will be going over those shortly. So, first decision, is the trait showing complete dominance, codominance, or incomplete dominance? And if the trait is showing complete dominance, which allele is dominant? Decision 2, is the trait autosomal or is it sex-linked? So, let's look at this. So, In cross 1, a gray male fruit fly was crossed with a black female fruit fly. The following results were obtained. So we have gray and black. So is it complete, incomplete, or codominance? Well, you can't have gray without black and white. So in this case, we're looking at incomplete dominance, and our letters would be B prime for gray, B for black, and B prime for white. And if you look, everything's pretty much equal between the females and the males. So to determine if it's sex-linked or not, you are going to see a huge difference between male and female. So in this case, there is no phenotype more associated with one sex. So if this was in fact sex-linked, then for instance, this black females versus gray females, or even right here, gray males, the black males. So for instance, this would be a huge number compared to the others. On top of that, you need to note this is basically a 1-to-1 ratio. Okay? So in a second cross, a black male fruit fly was crossed with a white-bodied female. The following results were obtained. 47 gray females, 53 gray males. So, what are the genotypes of the parents in cross 1? Remember, we have a 1-to-1 ratio. So if we take our Punnett square and we do what we think would be a 1-to-1 ratio, then what would we end up with? We're going to end up with capital B, capital B, capital B, capital B, and then again B with B prime, and B with B prime. And again, that is your standard 1-to-1 ratio. So we know here that we're looking at B with B prime for the gray, and B for the black female. So explain how you could tell whether the gene for body color was autosomal or sex-linked. And so in this case, what we would write is if more of one sex develops a phenotype, then the trait is sex-linked. That did not occur in this situation. Okay? Now, if you're not sure that you have the right information, then you can also double check by doing your second cross. And your second cross basically was a black male with a white-bodied female. So it would be your B prime, B prime, B prime, crossed with the capital B, capital B. So you can always do that to double check to make sure that you still end up with that 1-to-1 ratio. Okay, so problem 5. In a mating of a male Drosophila, I think I said that wrong, but regardless, it's a fruit fly, collected in nature with a normal female from a laboratory strain, 237 offspring were obtained. All of the offspring had normal eyes except one unusual male which had very narrow eyes. A series of matings is shown in the table below. Determine the genotypes of the individuals in the crosses. Okay, so we have in our first cross, we have an unusual male and a normal female. So, normal female, we already know XN, XN. So, here we have an unusual male with narrow eyes. So, but we have slightly narrow, we have very narrow. So in this case, this would be our kind of our incomplete. So it would be an X N prime with a Y. Okay? And then we could do that cross. So our first decision is just what we just did. Okay? First decision, we're going to decide that normal, we know we have normal, and we're going to do those in. Second is slightly narrow. And we're going to do N prime. And then we have very narrow, which is going to be N prime, N prime. Our second decision, is it sex-linked? Sex-linked. And we know that it's sex-linked because they basically told us it's a series of matings. And we see that the males are affected with the very narrow. So this is going to be sex-linked. So let's look at the questions. The most likely explanation for the appearance of the narrow-eyed male in P1 is what? Appearance of a hidden variability, occurrence of a mutation, consequence of laboratory culture, result of hybridizing a culture of the offspring at a higher temperature. Well, I would say that they didn't give us any information about that other than he was collected in nature. So we know that C is not it. And we didn't really talk about, hybridizing and temperature. They didn't give us any information about that. So I'm going to go with mutation. 2, assuming that a single locus causes the eye characteristics, the gene appears to function as, well, we know it's not dominant. We know it's not recessive. It is incomplete dominance. A gene typical of that found in nature. No. And so I'm going to go with an incomplete dominant allele. 3, from the breeding data, it is clear that the gene is physically located on which chromosome? Well, they didn't give us any information about chromosomes except X and Y, male and female. So we know that C, D, and E are not it. We also know that Y doesn't carry anything, so X is the correct answer. 4, why do the F1 males not show the genetic trait? The gene has a variable penetrance. Yeah, we didn't talk about that at all. The effect of the gene is blocked by the Y chromosome. That's not possible. The allele is blocked by its dominant allele. Well, we already know it's incomplete dominance, so that's not it. The trait is expressed only in alternate generations. Well, we see it in all. So, the answer would be E. 5, a promising approach to ascertain if a female with very narrow eyes could be obtained would be to cross normal with normal. Well, we know that's not going to happen. Cross the F2 males that are not normal with females that are not normal. That's a possibility. Cross F2 very narrow males with normal. Well, we already know that's not going to happen because we saw that up above. Cross the F2 slightly narrow females with normal males. Again, not going to happen. Back cross the F2 normal females with their male parents. And that could result in mutations. That wouldn't give us a result. So we're going to go with B here. Okay. So now that we've covered some of that, let's talk about chi-square. I know you've missed them. So using a chi-square analysis to determine if the percent of offspring you observe is a good fit to what was, of course, we are talking about chi-square, so it's what's expected. So a Punnett square is going to help us determine what numbers are expected. So problem 6 here. Use a chi-square analysis to determine if seed pods show complete dominance. A heterozygous plant with round pods is crossed with a wrinkled, and the offspring were counted. Out of 288, 159 had round and 129 had wrinkled. The expected numbers of 288 would be... Okay, so first thing is that we know that our expected numbers, we are looking at 50-50, right? So our expected numbers here would be 144 round and 144 wrinkled. We also need to determine what our genotypes are going to be. So wrinkled is going to be the recessive. So we are going to have wrinkled, and it said heterozygous, so we know this will be big R, little r. So if we do that cross, we're looking at 50-50. So let's go up here. Let's go to our chart. And our expected number, we just said, was 144. Because we're doing 50%. And that gives us, again, our 288. Our observed minus our expected in each case happens to be 15. So we're going to square that. Gives us 225. And 225 divided by 144 would be 1.56 for each one. So our value, our chi-square value is 3.12. So our degrees of freedom, if you go back up and look, we had two groups. So our degree of freedom is 1. Our critical value, remember we're going to use a 0.5, is 3.84. So our null hypothesis, we are going to say that there is no significant difference between what was observed and what was expected. So since the chi-square value is less than the critical value, we are going to accept the null hypothesis because we are 95% confident that the observed data was a good fit to what was expected. If, remember, the chi-square number is greater than the critical value, then we would reject that hypothesis. In this case, we are 95% confident that the observed 159 and 129 were a good fit to what was expected, the 144 and 144. Our chi-square value was lower than the critical value, meaning that we accept the null hypothesis and that we know that the seed pod shape does show complete dominance. Okay, that is the end of this lecture, and I will see you guys next time. We'll get into some more complicated crosses.